Hello, we are Dr. Zahn's group, and this is our mini presentation one. So the core of our project is using COMSOL to create models of cells. To first create a model, it is necessary to select what kind of cell we would like to create. Since we will be focusing on ellipsoid cells for our models, we must have inputs to generate the cell's geometry, such as the length of each semi-axis. We aim to keep the volume of our ellipsoid models consistent to our initial spherical model with a volume of about 524 micrometers cubed. In addition to this, we also build a cubical volume around the cell that will function as the extracellular matrix.
Once the geometry has been set, another factor, the material properties, must be set. We create a different material for the external medium, the contact impedance boundary, and the cytoplasm of the cell, with the contact impedance boundary functioning as a cellular membrane. Each of these parts have a different electrical conductivity and relative permittivity that must be specified using obtained theoretical values.
The electric field is the expression of volts per meter that is created by the electric charge applied via the cathode and anode ends of our simulation environment. We study the electric field to understand how the cell itself interacts with the applied voltage, especially in comparison to before and after it is electroporated.
The geometric setup and material properties of the ellipsoid cell with an applied electric field across it that was just described form the base of the single ellipsoid cell model. Modifications can then be made based on the specifications that are to be tested with our model. The orientation of the ellipsoid cell with respect to the electric field can be customized in order to view how orientation affects the degree of electroporation of the cellular membrane. Ellipsoids can therefore be oblate or prolate ellipsoids, as well as any angle between them. Also, multiple cells can be introduced at their desired orientation. This allows for customization based on how many cells are to be introduced and in what proximity they are to one another. The effects that these various cells have on each other and distortions they have on the electric field can then be studied.
We use MATLAB to help us find the regions of expected electroporation. We begin by extracting voltage data from COMSOL from points immediately inside and immediately outside the cell membrane. One of the advantages of using MATLAB for this purpose is that it is very easy to control the precision and density of data points.
Once the data is imported into MATLAB, we are able to use it to graph the transmembrane voltage versus the angle around the cell. This graph will show us the areas of the cell which are electroporated, as they will have a voltage greater than 0.5 volts or less than negative 0.5 volts. Using this data, we are able to go back to COMSOL and change the conductivity of those regions and thus generate a second transmembrane potential graph, which accurately portrays how the electric field distribution changes once a cell undergoes electroporation as according to the COMSOL model.
The simulation environment is created such that there is a uniform electric field of around 1 kilovolt per centimeter acting upon the cell based on accepted voltage values. The cell and electric field is then observed once target regions are defined as electroporated.
Once we have data from a multi-cell model, we are able to once again extract it to MATLAB, and using the transmembrane voltage graph, we determine areas of electroporation and generate a secondary graph which will demonstrate the shielding effect of cells within an electric field and how it impedes electroporation as a whole.
This is where our project steps converge, and we are trying to state that our model is fully optimized. Our main goal is to maximize the cell's electroporated surface area, and as mentioned, using the largest voltage we can, at varying orientations and maintaining a high level of viability. The end output of our project is a series of models displaying successful electroporation of ellipsoid cells.